Okay, so today is the fifth tutorial of the course called I Think Biology. It is a course offered on NPTEL by multiple professors from the Azim Premji University. I am Geetika Agarwal. I am a PhD scholar at the Center for Ecological Sciences in IISc Bangalore, and I will be taking this course today. And so, first of all, I will summarize what happened or the topics that were broadly covered during this week of the lectures. And this lecture was mostly talking about genetics and the Human Genome Project and some genetic disorders and mutations and things like that. And uh, then there will be a short Q&A session at the end of this tutorial. Okay, so moving on to contents of this week or the topics that were broadly covered, the lecture started out by talking about genetics and some introduction to genetics and what was the role of Gregor Mendel in genetics and why is he called the father of genetics, and the discovery of the chromosome and uh, the DNA, and then talked about the Human Genome Project and how it led to the sequencing of the entire human genome. And then we discussed a little bit about genetic mutations and how that leads to genetic disorders, and slightly about the how model organisms or transgenic model organisms are used in research to study more about or to understand more about uh, genetics and inheritance. So, as you all might know by now, or as we've been discussing in the past lectures during the past weeks, that all life is encoded for by the DNA. So, DNA is basically. Hi, am I audible? Uh, Hi, so today is the fifth tutorial of this course, I Think Biology. And I was just saying that the contents covered during this week were mostly about genetics, and they talked a little bit about the experiments conducted by Gregor Mendel and how they helped with understanding more about how inheritance happens and how these traits are taken up from the parents to the progeny. Then they discussed the Human Genome Project and how the human genome was sequenced, about genetic mutations, genetic disorders, and transgenic model organisms and how they are used in research. So, as we've discussed a lot in the multiple lectures, that all life is encoded for by DNA, in the sense that DNA forms this basic instruction manual in all of the living organisms, and we are all united and we share the same language in which this manual is encoded, because this manual is encoded in the language of these nucleotide bases A, T, G, C. And that is, it stays the same across all the living organisms, starting from bacteria to plants to animals to humans. All right. So, and much of the biology or much of the study that we do, even if it's on other organisms, it is very well applicable on humans because of the fact that we all share the same language or the same DNA encoding, right? So, if you learn something else in other organisms, it is very well applicable to humans as well. And DNA, in that sense, you can think of it as a way of storing information, because we're storing the information in the code, in the sequence of these nucleotide bases. So DNA helps you store this information on how the cell is to be run and what are the proteins to be encoded and things like that. And this information, it can be exchanged across organisms and across species. So that is what biotechnologists do more often. They or the researchers that construct, these organisms which are genetically modified. So this is how they exchange information between these organisms. So you take some genes, for example, say you, for treating diabetic patients, you want to create huge amounts of insulin, but creating synthetic insulin is actually very costly. So what they do is they take out the gene that encodes for insulin and insert that into bacteria. And now, because bacteria have a very small reproductive or doubling time, so they can double in 20 minutes. So you get a huge amount of insulin out of, you can get huge quantities of insulin out of these genetically modified bacteria. Okay, so that was the basis. And what is inheritance and what is heredity and how are they two different? So, what are these two terms, inheritance and heredity? So, when you look at inheritance, inheritance is a process and heredity is a phenomena. So, heredity is a phenomena which occurs by this process which is called inheritance. So, there's a very subtle difference between the two of them. And inheritance is the specific way in which traits they are passed on from one generation to next, which means from parents to the progeny. So, inheritance is the specific way in which these traits are being passed on, and it involves the study of the transmission of traits, and it is important for us to know which that some of them are dominant traits, some of them are recessive traits, and we'll discuss about these in more detail later when we talk about Gregor Mendel's experiments. So, but inheritance is this process, or it's the specific way in which the traits are being passed on from one generation to the next. Heredity is the biological phenomena through which these traits get passed on from parents to the offsprings. So, heredity is the phenomena that occurs by the process which is inheritance. So, keep that in mind. Then, so if you talk about selective breeding, so it was not like only recently or after the scientists actually studied inheritance, is that so it is not so that humans, got to know about breeding only after, or got to know about inheritance only after the scientists actually studied it and gave us some theories about them. Breeding has been practiced since a very long time. So early humans have been indulging in breeding. They have been selectively choosing for traits in animals which make them more docile, which make them more beneficial to humans. So domestication happened very early on in the human societies. So selective breeding is also known as artificial selection. It's a process by which humans they use animals or they use plant breeding to choose for desirable traits in those organisms. For example, to choose for more beef, if we're talking about cows, to choose for a higher quantities of high milk producing cows, or to choose plants that are more resistant and that give you higher crop yields. So selective breeding has been happening for a long time now. And if you so yeah, and if you look at the time scale since when this has been happening, uh, so as early as one second, sorry. So I was just saying that as early as 20,000 BCE, humans have been indulging in domesticating wild animals. So we have been practicing selective breeding for a long time now. And so it's not that we were not aware of selective breeding and how the mechanism works, it's just that we didn't know the science behind it. And that is where Gregor Mendel comes in. Gregor Mendel is also known as the father of genetics because of the huge amount of because of his efforts and because of what he gave to the community, or what understanding he left behind. So he's called the father of genetics. And to give you a brief background, Gregor Mendel, he does not come from a very wealthy family, but he really wanted to continue education. So he chose a monk life because in that time, if you become a monk, your education was sort of covered for, it was free. So he studied mathematics and physics, and then he came to Austria, where he started working with other organisms, and he began his research with mice and later switched to honeybees, plants, and ultimately he settled down on garden peas as his primary model system. So he had this backyard garden in which he had these pea plants, and he started studying those pea plants for his experiments. So it was between 1856 and 1863. It was in these 7 years when he conducted these hybridization experiments on the garden peas, or the scientific name was Pisum sativum, as you can see in this image. So he studied garden peas, and he carefully chose some very distinct characteristics of these peas, which showed up in the phenotype as well, so that it made it easier for him to track uh, how heredity is working, how genes are being inherited. so that it became easier for him to track which characteristics are inherited from parents to the offspring. And then he conducted some cross pollination, some artificial pollination experiments on these pea lines. So to give you and eventually, from his experiments, from his outcomes, he postulated the Mendel's laws of inheritance. So you might already be aware of the Mendel's laws of inheritance. We are all taught this in our high school, but we will go through them again once today. So if you look at what were the traits that were chosen by Mendel, what were the traits that he was looking at in these pea plants? So he looked at these seven traits. First was the seed shape. So either the seed could be round or the seed could be wrinkled. So it could be a round seed or a wrinkled seed. Then he looked at what was the color of the seed. It could either be distinctively yellow or green. What was the flower color? It could either be purple or white. And then he chose the pod. He saw that the pod shape could either be inflated or a constricted pod. The pod color could be yellow or green. The position of the flower could be axial or terminal, and the stem height could either be tall or dwarf. So these were these seven characteristics or seven traits that Mendel chose to include in his study experiments. And what he did with this was, number one was that he developed true bred strains. So what are these true breeding strains? So these true breeding strains, they're actually strains which, when pollinated either with themselves or with other true breeding strains which have these same traits, they will always produce progeny that has the same traits. So the progeny will always be identical to the parents. So if you have, for example, if you have a true bred strain with purple flowers, then if you keep pollinating it with itself, if you keep self pollinating this plant, it will always give you uh, progeny or with purple flowers. Similarly, if you have a tall plant or if you have a dwarf plant, if it's a true bred, then if you keep selfing these strains with themselves, if you keep self pollinating them, they will always give you the same phenotype back. So a true bred tall plant will always give you tall progeny, and a true bred dwarf will always give you dwarf progeny. Okay, so this is what he started with. So this was his first step, that he made these true bred strains. So he kept self pollinating these plants over and over and over again till he reached a point that they no longer gave you any other progeny apart from that true bred strain. Okay, and then so progeny always carried the same trait as the parents. And he established true breeding strains for two features, such as purple flowers and white flowers, or tall plants and short plants. So the two traits that he was comparing, or the one trait that he was comparing, and the two phenotypes in that trait, for example, if he was comparing flower color, then purple flowers and white flowers were the two strains, or the two strains that he developed. And if he was comparing the plant height, then tall and short plants were what were the strains that he developed. Okay, so he actually came up with strains which were distinct only this one characteristic. So if you were talking about these two pea plants with purple flowers and white flowers, they will be identical in all the other traits. For example, they will be tall, they will have constricted pods, they will have yellow colored pods. Everything will be the same. The only difference they will have is that one of them will have purple flowers and the other will have white flowers. And same for the other condition. If you're talking about the plant height, all other characters will be the same, only they will be different in this height characteristic, that you will either have tall or short plants. Okay, so this is where he started at. Then what he did was, now he cross pollinated these two purebred strains. Now he has two pure breeding strains. One will give you purple flowers, one will give you white flowers. Then he cross pollinated them. And what is cross pollination? As you might already be aware, so he manually he collected the pollen from say the purple flower, and manually put it, or uh, uh, transferred it onto the female, which is the white flower here in this case. So if he's using the male parent as the purple flowers and the female parent as the white flower, then he manually took the pollen from the purple flower and then put it on the stigma of the white flower. Or other way around, it took the other way around would be, and in order to prevent selfing, he would actually cut off the anthers of this of the female plant, so that it does not get self pollinated before he conducts cross pollination. Okay, and if it was the other way around, then he would have cut the anthers of the purple flower and he would have used the pollen of the white flower and deposited on the stigma of the purple flowers. So this is how he crossed the plants. Okay, so he manually took the pollen from the anthers of one plant, which was considered the male parent, and then he deposited it onto the stigma of the other flowers, which were considered as the female parents. Okay, so after doing so, what he saw was, after this cross, the seeds that he got, so in the first generation, so the seeds that he got after this cross, the all the flowers were purple. So if he planted those seeds and then saw the outcome of the plants that sprouted from those seeds and the flowers that came on those plants, they were all purple. So that means that, oh, what he expected, or what was expected at that time, was something called blending inheritance, in which they always thought that if a tall and a short parent, they mate and they have progeny, then the progeny will be medium heighted. So in this scenario, he thought that if a purple and a white flower are being mated and then you're seeing the progeny, he would get some light purple flowers. But that is not what he saw. What he saw was that the in the first generation, all the flowers were entirely purple. And now this is the first generation, is called the first filial generation, or the F1 generation. Then what he did with them was, with the first filial generation, he selfed them. So these purple flowers, in them, he prevented cross pollination by covering a bud with a polythene bag, so that it does not get any external pollen, and so that selfing could happen. And so from the same anthers to the same stigma, if the pollen goes to the same flower, or from the same flower's anthers to the same flower stigma, then there's a self pollination. So by self pollination, then you get the second filial generation. And in the second filial generation, he saw that, okay, some of the plants came out to be with purple flowers and some of them had white flowers. So he was very surprised how this white trait, which was lost in the first generation, now reappears in the second generation. So that was very surprising. And one important thing to note here was the ratio. He saw that out of four plants, roughly three of them had purple flowers and one of them have white flowers. And to make it very clear, here we're talking about a large number of plants. We're not just talking about two, three, or four plants. When he worked with plants, there were like hundreds and thousands of pea plants that he was working with. So these are like rough ratios. These are not like, not absolutely out of four, three will be this and one will be that. It's when we're talking about on a large scale of thousands of plants. Okay. So this is again to show you how this happened. So there was a cross between two true breeding pea plants, one with violet or purple flowers and one with white flowers. When this hybridization happened, you got the first filial generation in which all the progeny had purple flowers. Then when selfing of this filial generation happened with itself, then after self fertilization of these hybrid plants, you got the F2 generation, in which you got back purple and white, both of these flowers. And violet flowers were like, out of 1,705 were violet and 224 had white flowers. So roughly a ratio of 3 is to 1 was followed. So this is how he conducted his experiments. To give you another idea of tall versus short, and here you can see that I've written capital T next to tall and small t next to short. So this is to tell you that, and I will come to this later, that after his experiments, Mendel thought that, okay, so every organism or every individual pea plant, it carries the characteristic, it carries a pair of factors. So at that time, he did not know about genes, he did not know about chromosomes, he did not know about DNA. So he came up with this term called factors, and he said that every individual carries two factors which encode for that gene. So for a true bred tall plant, both these factors will be capital T, which means both of these factors will encode for tallness. And for a true bred short plant, both of these factors will be small t, both of them will code for a short plant. But in a hybrid, it could be a capital T and a small t, in which one of them encodes for tallness and one of them encodes for shortness. So that is why I've written it here. So, again, just as in the case of violet and white flowers, here in the parent generation, you have a true bred tall plant and a true bred short plant. From this, you perform a cross between these two, and in the first filial generation, you see that all of the progeny is tall. Then, after selfing this progeny of the first filial generation, you get the second filial generation, in which you see this rough ratio of 3 is to 1, where three of them are tall and one of them is short. So this is so out after seeing these experiments, after seeing these results, Mendel came up with dominant and recessive traits. So what are dominant traits? He said that the traits that appeared in the first filial generation, they are dominant traits, and the ones that were hidden in the first filial generation but reappeared in the second filial generation, they are recessive traits. So in this scenario, tall is a dominant trait and short is a recessive trait. So when tall and short come together, so if they form a progeny which has a capital T and one small t, then the capital T will overpower the small t, and shortness will not express itself, but the plant will be tall because the tall gene will overpower, or the tall factor will overpower, and so all of these will be tall. But when you come to the next one, the next generation, you'll see that shortness comes again. So short is a recessive trait, tall is a dominant trait. When it comes to purple and white flowers, purple flower is a dominant trait, white flower is a recessive trait. So in the F2 generation, there were always about 3 plants that showed the dominant trait, here tall, and there was one plant for every 3 plants that showed the recessive trait, which was like short. So it making a rough ratio of 3 is to 1. Okay, so this is what he saw, and this is what he came up with, that there are these dominant and recessive traits. So let me discuss the Punnett square. Punnett square is a way in which you can actually calculate what the phenotype will be from the genotype. So if you're talking about the first filial generation from these tall and short parents, so in the tall parents, as I said, there is capital T, capital T. So these are the two factors or the two alleles that are coming from the parents. So um, from the factor, for example, say the gene is height, then capital T are the two alleles. So that code for tallness in that gene. And here small t are the alleles of the short parent that code for a short height. So when capital T, small t, so this is how you create a Punnett square, in which on one side of the box you write capital T, which is the alleles coming from one parent, and on the other side of the box you write small t, which is the genes coming from the other parent, and then you make this box, and then you divide it into 4 sections as is divided here, and then you just make these combinations. So one capital T coming from here, small t coming from here, same capital T, small t, capital T, small t, capital T, small t. And this progeny, which is one capital T, one small t, all of them will be tall because capital T is the dominant trait. So it takes over, and this is only what gets converted into the phenotype, or what shows up in the progeny. And small t is not expressed as a phenotype. And when you look at the second filial generation, now if you cross a hybrid with a hybrid, like in the second filial generation, you have all, so in the first filial, you had all capital T, small t. So if you cross a capital T, small t with a capital T, small t, you see that you get a capital T, which will be tall, a capital T, small t, which will be tall, a capital T, small t, which will be tall, and a small t. And this will be short. And you can see here from the Punnett scale that for every 4 offsprings, there are 3 which give you the dominant phenotype, and 1 which gives you the recessive phenotype. So you can use the Punnett square to actually calculate the phenotype if you know the parent genotypes. Okay. So now let's talk about something which is not discussed in the lecture, which is the monohybrid versus dihybrid cross. So I'll just explain it a little bit to you guys. What is a monohybrid cross and what is a dihybrid cross? So to give you some basic differences, in a monohybrid cross, it is a cross between two individuals, in which we are only considering one contrasting trait as a time. So what we discussed before, talking about flower colors, talking about height, those were monohybrid crosses. Because when we're talking about flower color, we were only talking about flower color and nothing else. And we're talking about height, we were only talking about height and nothing else. So it is a cross between two individuals in which you're only taking one single trait at a time. ठीक है. And a dihybrid cross is a cross in between two individuals when you are taking two contrasting traits at a time. So when you're considering two contrasting traits at a time. So if you're comparing individuals which have purple flowers, okay, and are tall, and you are doing this cross against individuals which are recessive for both, which have white flowers. So with a small p, I'm showing white flowers because capital letters are shown are used to denote the dominant traits, and small letters are used to denote the recessive traits. So if you have this cross, where you are crossing plants which have purple flowers and are tall with plants that have that are short and have white flowers, then that's called a dihybrid cross, because you're considering two contrasting traits at a single time while crossing two individuals, right? So this is the difference between dihybrid and a monohybrid cross. So in a monohybrid cross, it helps you to study inheritance of a pair of alleles. So it will help you to study inheritance of two alleles, which is say capital P, or or like capital T small t. In this, the dihybrid cross, as you can see, will help you study the inheritance of a pair of traits. So you're looking at a pair of alleles, two pairs of alleles, sorry. So one alleles for the flower color, and one pair of alleles for the height of the plant. So you're looking at two pairs of alleles. So the phenotypic generation in the F2 for a monohybrid cross is 3 is to 1, as we've already discussed here in the last slide. So here you get a ratio of 3 is to 1 in a monohybrid cross for an F2 generation. So, but what is the phenotypic ratio for um, for an F2 generation of a dihybrid cross? So this is how you calculate the phenotypic ratio of a dihybrid cross. Again, you're using a Punnett square here as well. So it's a dihybrid cross. So here they are talking about yellow pods, which are or yellow seeds which are round, and these are green seeds which are wrinkled. So yellow and round are dominant traits because they are denoted by capital letters, and green and wrinkled are recessive traits. They are in small letters. Okay, so they, if you cross these, then all of the progeny will have capital Y, capital R, small r, and they will be dominant in the first filial generation. So this is the F1 generation. Okay, this is the this is the parent generation. uh, this here is the parent generation, and this gives rise to this F1 generation. And if you self the F1 generation, then you get this F2 generation. In F2 generation, the gametes that can come from the heterozygous parents is, you can either get a capital Y for yellow color, or a capital R for a round seed. You can get a capital Y for yellow color, small r for wrinkled seed. You can get a small y for green color, and a capital R for round seed, and a small y and a small r for green and round wrinkled seeds. Okay, so these are the four combinations, and when you combine all of this, this is what you get. And here you see that here um you will get, if you compare the ratios of the different phenotypes, you see that you get a dominance of yellow and round seeds are in the number 9. So the proportion is 9. And if you have yellow and wrinkled seeds, 3, green and round seeds, 3, and green, or like both of them being totally recessive, is 1. So out of these 16 outcomes, so here you have a 4 by 4, right? So out of these 16 outcomes, you get 9 plants with yellow and round seeds, you get 3 plants with yellow and wrinkled seeds, 3 plants with green and round seeds, and 1 plant with a green and a wrinkled seed. So this is what a dihybrid cross is. I hope it is clear to all of you. Moving on to coming to the outcomes. So, what did Mendel find out? So, Mendel found that each parent, or Mendel concluded basically, that each parent carries a pair of factors, which we now call genes. But he didn't know that genes, what are genes, so he called them factors. So, he found that each parent carries a pair of factors that control the trait. So, he said that there are these factors, and every parent has a pair of factors that control the trait. Okay? So, every trait is controlled by factors, and these factors, they can be inherited by the offspring, and their inheritance is independent of each other. So these factors, they are passed on to the offspring, and that is how, inheritance is, or heredity is happening, and their inheritance is independent of each other. So each factor comes in different versions. So every factor can come in different versions. So we have, say, so two factors means, for example, tall and tall. So there are two of these copies. So there are two factors, and they can come in different versions. So it could be tall, it could be short, it could be medium. So all of these things, right? So it comes in different versions, and these different versions is what Mendel called heritable elements, which we now know are alleles. So we now know that there's a gene that encodes for a particular phenotype, and that gene can be in different forms, which are called the alleles of the gene. Okay, so at that time he didn't know that, so he called the genes as factors, and he called these alleles as the heritable elements. So there are different versions of the factor which he called heritable elements. And as I said, Mendel was unaware about the existence of genes and chromosomes. And then he went on to propose the laws of inheritance. So let's discuss what are these different Mendelian laws of inheritance. It's very important if you're talking about genetics, if you're discussing about genetics, or if you have an exam about genetics, it's very important to know these Mendelian laws of inheritance. So what do they talk about? So first of all comes the law of dominance, which is the very basic law in which we say that each factor, it comes in different versions, or it has different alleles, and a dominant allele is one that can hide the recessive allele, and the dominant allele is the one that determines the organism's appearance. So that is why in the filial 1 generation, in the F1 generation, we saw that the dominant allele always completely overshadowed the recessive allele. So if you have a tall plant being crossed with a short plant, you always got tall plants in the F1 generation because all of them had the dominant allele, capital T. So a dominant allele hides a recessive allele and it determines the organism's appearance. So the short allele could not do anything about the animal organism's appearance because all the pea plants were then tall. So it's the tall allele, which is the dominant allele, which determined the organism's appearance. The next law is the law of segregation. In this law, Mendel said that when an organism makes gametes, each gamete receives just one factor copy. So, when an organism is making gametes, each gamete will just receive one factor copy from that organism. So, that is how the Punnett square comes into being, right? You either get so if you are coming from a hybrid parent, you'll either get capital T or you'll get small t. So, each organism gets one factor copy and which is selected randomly. So, whether you're getting a capital T or a small t is entirely random. So that is selected randomly, and this is called the law of segregation. So an organism can either end up getting capital T or a small t. There is not a way in which it's being selected. It's mostly a random process, is what Mendel said. And then the last of the Mendelian laws is the law of independent assortment. In this one, Mendel says that the alleles of two or more different factors, they get sorted into gametes independently of one another. So the alleles are not being sorted based or they're not dependent on each other for being sorted. So for example, the alleles for height are independent of the alleles for flower color, are independent of the alleles of pod shape, are independent of the allele of seed color. So it's not that if one of the pea plants has yellow seeds, it will always have purple flowers. If one of them has green seeds, it will always have short height. So that is not the case. All of these alleles are getting independently assorted. Their assortment into the gametes is not dependent on each other. So they are independently assorted. So in other words, the allele a gamete receives from one factor does not influence the allele received for another factor. Okay. So I hope this is clear to everybody. And yeah, so these are the three laws or the Mendel's laws of inheritance. So these three laws: law of dominance, segregation, and independent assortment. Moving on to how was it figured that chromosomes carry traits. So till now we have discussed कि Mendel did not know that there are genes and there are chromosomes. Then how was it figured that chromosomes carry traits? So it was actually so there were these two people, Boveri and Sutton, and they came up with a chromosome theory. So they also did not prove anything scientifically, but they had this theory, and their theory was that genes or the factors that Mendel talks about, or these genes, by that time they knew that they were genes, they are found at specific locations on the chromosome. So they knew about the presence of chromosomes. They had been looking at how mitosis and meiosis is happening. So they said that these genes that Mendel is talking about, they're present on specific locations on these chromosomes only. And the behavior of chromosome during meiosis can explain Mendel's laws of inheritance. But they had not actually proven this theory of their own. They came up with this chromosome theory, but they had not proven anything. Then comes Thomas Hunt Morgan. You can see here, this is Thomas Hunt Morgan, and he used to work with fruit flies. So this is his lab. So you can see there are these huge numbers of vials in which he actually used to breed Drosophila melanogaster, which is the fruit flies. Sorry for the typo. should be small m. As we know in the scientific name, species should always be written with a small letter. Yeah. So he worked with Drosophila melanogaster, uh, and in around 1908. So that was the timeline in the 1900s, in early 1900s. So he had a fruit fly collection, as you can see in this image. So he had these all these vials which are full of fruit flies, and he was breeding them. And why he chose fruit flies is because they are cheap, because they can be stored in large numbers, because they are small organisms, and because they have a small breeding time or small doubling time. They will you know their generation time is small, so they will develop faster, so you will be able to follow their lifespan and follow how diseases or things spread in them. So he had this fruit fly collection, and he had been breeding them, and he wanted to look at mutants. So it was only after 2 years that he saw a mutant male fly, and he saw, out of all these vials that he has, he found that one male fly which had white eyes, and so that was a mutant. So it was different from the wild type, which was the red eyes normally seen. So this is what the wild type fly looks like. The wild type male, it has red eyes, you can see very clearly. But the mutant fly had white eyes. So Morgan had finally found his mutant fly. Now what he did was, he took the white-eyed mutant, and then he crossed it with the wild type females. So this is the female. So you can see from these diagrams that in, so how did he know that it was a male? From these cartoons, you can see that it's very easy to distinguish males from females in Drosophila. One of them is that males are smaller in size, and the other thing is that males have this black spot on the end of their abdomen, or the end of their tail, but females lack that. Okay, there's no black spot. So yeah, but he crossed these mutant white-eyed males to the red-eyed females, and what he saw in the first filial generation was that, wow, all of the progeny has red eyes. So all the males and all the females, all of them have red eyes. Then what he did was, he crossed these only, he crossed these red-eyed females with these red-eyed males which were received in the first filial generation. And then what I saw, to his surprise, was there were if you look at two females, they were they had red eyes. Then one out of these four, one male had red eyes and one male had white eyes. So that is the ratio that he got. 2:1:1. So he started out with a parent generation in which he took one mutant male with white eyes, and he crossed it with a wild type red-eyed female. Then in the first filial generation, the all the progeny, irrespective of sex, had red eyes. And then in the second filial generation, when he looked at the progeny, it followed a ratio of 2:1:1, in which 2 is for the females, which are all of them were red-eyed, and 1:1 is the ratio of the males which had red eyes to the males that had white eyes, or which were mutants. Okay. So then, so what he concluded was that white eye is a heritable trait. It is being passed on from parents to offsprings. And very interestingly, it is only being observed in males. So he did this experiment multiple times. He just did not do it once and conclude by that only. He did this experiment multiple times, and he observed that, oh, okay, this trait is only found in males. So he then came to the conclusion that it would be linked to the sex chromosome. So by then we knew about the existence of chromosomes. We just did not know that they are the things that are carrying the genes, and they are responsible for inheritance or heredity. Okay. So he now concluded that, okay, fine. This link, this white eye gene, or this eye color, is actually being linked with the sex chromosome. And as we know, XX is what is found in females, and XY is what found in males in Drosophila as well. So this is what the Punnett square for this would look like. So if you have a female which is red-eyed, so if X is the chromosome, or it's an X-linked trait, and on the X chromosome you have this wild type gene which is W plus, which is being denoted by W plus, which is wild type, and the male is XW, and the other chromosome is Y. So he's saying that this gene is linked to the X chromosome. Okay. So why is it being called W or XW is because whenever, so that's just a way or a convention of naming things. So if you come across a mutant, you name the gene to the corresponding to the mutation. So because it's a white-eyed mutant, so the gene was named X and W, and the wild type is then named with X and W and then a plus next to it, because it's a wild type. So that is just a convention or naming convention that's followed. Okay. So when you see the Punnett square of this cross, you see that in the first generation you get females which get one X from their mother, which is the wild type, which carries the wild type eye color, and one X from the father, which carries the white eye mutation. Then both the males, they get their X from the female. So that's why both the males actually have a wild type eye color. They both have red eye color because they got the X from their wild type mother, female parent. Okay. And now both the females are actually hybrids. They have one chromosome which carries the gene of white eye, and one chromosome which carries the gene of red eye. But because red eye is the so in this case red eye would be the dominant gene, right? So in this case red eye would be the dominant allele, sorry. Yeah, because it is overshadowing the white eye, and the females still have the red eye color in the progeny, or the phenotype is still red eye. So red eye is the dominant allele. So when you move next, now when you move to the F2 generation, so in the F2 generation you are crossing the male and the female received in the F1 generation. So the males which are received in the F1 generation are XW plus Y. So they are wild type. They have the wild type X and Y chromosome, and the females are hybrids. So if you cross them, you get this progeny in which one of the female is homozygous dominant. The male has a dominant X, one of them, because it gets its wild type from the female, and one of them gets the white eye mutation allele from from the female mother, and that is the white-eyed male. And in the female progeny, you get females which are homozygous dominant, and you get females which are hybrid. But even in the hybrid, because red eye is the dominant trait, they still show the red eye phenotype. So that is how you get the ratio of two females which are red-eyed, one male which is white-eyed, and one male which is red-eyed. So this is how you get that ratio of 2 is to 1 is to 1. So I hope this is clear. These Punnett squares very clearly explain what is actually going on in these experiments. So this is how Thomas and Morgan found that, oh, okay, that eye color is actually an X-linked trait in Drosophila melanogaster. Now we have talked so much about um, traits and traits being passed on in generations. So let's discuss what exactly is a genotype and what is a phenotype. So when you talk about a genotype is the genetic code or the genetic makeup which is present in the DNA of an organism. So what is the genetic code coding for? So that is what is genotype. Genotype is the genetic code in the which is found in the DNA of an organism. So it is the sequence of nucleotide bases or the gene. And the phenotype are the physical characteristics that can actually be observed. So for the case of Drosophila, the presence of that allele on the X chromosome is the genotype. But the phenotype of seeing either red eyes or white eyes is actually what is the characteristic that we physically observe. So that is what is called the phenotype. And so there are two approaches to studying genetics. One of the approaches called a forward genetics approach, in which you actually look at the phenotype and then you try to determine the genotype. So you know what is the phenotype. So like the approach that um, Morgan took. He knew the phenotype of white eyes and the red eyes, and then he tried to determine what is the genotype, where is it coming from. And the second approach is actually reverse genetics, in which you know what is the genotype, but you're trying to figure out what phenotype it leads to. So these are the two approaches that are employed by geneticists. So um, that is what is forward and reverse genetics. Okay. Moving on to this very big endeavor taken by the science community or the biological community, which is called the Human Genome Project. You might already be aware of this. Or the Human Genome Project, also called the HGP, it was an international scientific effort. So all over the globe, scientific effort. It was not limited to one country or one continent. Okay, it was an international scientific effort, and their goal was to sequence the entire human genome for the very first time. It was launched in 1990 and completed in 2003. Now, this is also a false statement because it was not even completed in 2003. In 2003 also, we only knew about 85% of the genome. It was only in 2022, which is like 3 years back, that we actually got 100% of the human genome. So if you count it from 2022 to 1990, so that's been 32 years of the process to actually sequence the entire human genome. But fine, most of it got sequenced by 2003, which is also a 13 years of effort. So, Human Genome Project was an international scientific effort and they wanted to sequence the entire human genome. Launched in 1990, completed in 2003. So, where did they get the DNA from? They got these 20 volunteers in New York. They denote, they not denoted, sorry, they they donated. So sorry. Yeah. So they donated their DNA to this project, and actually, out of all of these 20 volunteers, mostly the DNA or the final DNA that was found, the human genome that was sequenced, came mostly from one of the volunteers. But yeah, they got DNA from 20 volunteers, and they used DNA sequencing techniques to get the sequence of the entire human genome. And what they found, and it cost them $3 billion. So at that point, it cost them $3 billion. Today, it costs roughly around $1,000 to get DNA sequenced. So you can see how the cost has been cut a lot. Has is now much less than it was at that time to sequence one human genome. So, but that time it cost them $3 billion, and they found that the human genome is made up of 3.2 billion base pairs of DNA, and that the human genome is very similar between individuals. So they found that in total, the human genome is made up of 3.2 billion base pairs, and the human genome is very similar across individuals. So if you look at the genome between different individuals, much of it is entirely similar. Only like 1% of it is different between different individuals. And us humans, we have around 20,000 protein coding genes. This is also still an issue which has not been resolved, or we still don't know what is the exact number of protein coding genes or exact number of genes, but we have roughly 20,000 protein coding genes in our DNA. So these are some of the findings of the Human Genome Project. And if you compare the genome sizes of different organisms, you see that in these single celled organisms like bacteria, fungi, these mycoplasma, E. coli, you have the DNA sequence or the number of base pairs, nucleotide pairs, and around in the order of 10 is to 5 or 10 is to 6. So millions of them. Then you move on to say protists and plants, you get them in the range of 10 is to power 8. For plants, it has a huge range. It goes for as big as 10 is to the power 11. Insects span the range of 10 to the 8 to 10 to the 9. Mollusks also similar range. And mammals and humans fall here. As I said, we have 3.2 billion base pairs, so 10 to the power 9. Birds also around 10 to the power 9. So earlier, humans used to think that we are like the superior beings, we are better than all the other organisms. So our genome will be very big and it will be very complicated compared to other organisms. But later, scientists found out that that is not the case, actually. And compared to us, there are many other like marine organisms like these bony fishes, there are sharks, there are these plants which have much more or much bigger of a genome than we carry. But so that is not something that can tell you what is a superior organism or not. So so this is just like a rough comparison of how big the genome sizes are in different organisms. So, what is the significance of this Human Genome Project, of this huge endeavor, this international endeavor that scientists undertook in 1990 and took 13 years to sequence the genome? What is the significance of this? So, one of them is that it has now accelerated the study of human biology. Now that we know about so many genes, we can then very easily know about how are these proteins being sequenced, which gene is responsible for sequencing which protein, how is the disorder happening, uh, things like that. So it has accelerated the study of human biology. It has improved the practice of medicine because now we know where all of these disorders are coming from and what is responsible. It has helped identify the genetic predisposition to diseases. So if you know, like the phylogenetic tree of a particular human, if you, for example, if you sequence the genome of a person and then their parents and then their siblings, you can see if they have certain genetic mutations and if they might be Or if the parents carry, say, genes for diabetes, then the offspring might also have be predisposed to getting diabetes or getting cancers and things like that. It helps you tailor medications to individuals. So if you sequence the DNA of a particular individual, then you can directly give them medications that target their specific genome and their specific genes that are required to be targeted to treating their diseases. So you can get tailor-made medications. It helps family understand what are their different inherited conditions, and it led to this new ethos of data sharing in biomedical research. So because it was an international effort, data was fully being shared across countries. So it was this huge effort in which everybody came together, all the scientists came together, and the data was being shared, and there was no, you know there was no fight against this, that okay, USA is not going to share the data to other countries, or other countries are not going to in put in efforts into all of this. So it led to this new ethos of data sharing in the biomedical research, and it helped usher in a great emphasis on ethics in biomedical research. So it is very important when you're talking about genes and genetics. It is very important to keep the ethics of it in mind as well, because there are certain scientists are now actually, in this day and age now, because everything has been so like uh, technology has progressed so much that you can actually come up with uh, zygotes within a petri dish. You don't really need uh, things to actually happen physically. You can actually come up with zygotes, you can come up with babies, everything in a petri dish itself. So you can, that is why parents can actually have things like, there are things like designer babies, which means that you can alter your child's DNA sequence in a way to make it fulfill whatever you want it to be. So you can like ask the scientists to put in genes for tallness. You can ask them to take out genes which cause genetic disorders. So as long as you're treating things that deal with genetic disorders and things like that, that is fine. Then maybe, I don't know if that is also fine, but yeah, that is thought to be still ethical. But a lot of these questions stem from biomedical research. Now that we have so much power, should we be allowed? Now that we can alter the DNA of other organisms, we can alter the DNA of humans, we can alter the DNA of mice, birds, anything. But should we be doing all that? So this it leads to a lot of these questions which should be considered by budding scientists who are actually entering this field and who want to pursue this as their career. Okay. Okay, now there is a short video about Sanger sequencing. This is the sequencing method that was used to sequence the human genome in the HGP project. So this is a short video which will very nicely explain with animations how Sanger sequencing is carried out.
Sanger sequencing has been the foundation and gold standard of DNA sequencing since 1977. While new sequencing technologies have emerged since, Sanger is still an invaluable tool for all kinds of labs. The basic steps of Sanger sequencing are simple. The reaction involves template DNA, a primer, DNA polymerase, nucleotides, and fluorescently labeled dideoxynucleotides. First, the DNA is denatured and then cooled to allow the primer to bind. The DNA polymerase enzyme then lengthens the strand according to the DNA template until it randomly places a dideoxynucleotide. The dideoxynucleotide is missing the hydroxyl group, which allows the next nucleotide to bind. So once it is placed, the chain is terminated and will not grow any longer. The color of the fluorescent dye on the dideoxynucleotide represents the last base which was added to that fragment. This reaction is repeated until the sample contains the fragments of all different lengths, each terminated by a fluorescently labeled dideoxynucleotide. When the fragments are separated by size, the dyes can be read in order to give the sequence of the template DNA.
Yeah. So
Sanger sequencing has
this
been
was
the
a
foundation.
short video explaining Sanger sequencing and how it's carried out. So basically, you have this template DNA, which is actually placed on these things called beads. so that it can be, mounted on something and held in a location. And then, as mentioned, this template DNA is, the strands are separated, converted into single stranded DNA by heating it up, and then you bind primers to that single stranded DNA, and the primers then, by the presence of DNA polymerase, they get multiplied, and at different locations it places these dideoxynucleotides, which are actually fluorescently tagged, but they stop the increase in length of the primer. So from the template, at different lengths, you will have this dideoxy, and from these different lengthed strands that you get as a result, you can see that where which DNA comes. So, for example, you have a strand which is like two nucleotides long, and in that you can see that the fluorescent sequence is showing you the color for A. For example, A is tagged with green, so then you know that here adenine comes. And then say you have another one which is 4, and there you have C, and then another one which is 6, and there you have G, and then you have one which is 3 bases long, that there is T. So now you can figure it out. So you have A and T, and then C, and then something, and then G. So like that they figured out how, like that they sequenced the entire DNA. It was a long process, and now there are new and better sequencing methods, for example next generation sequencing, which can actually sequence huge amounts of DNA in one process itself. So they have cut down, as I said, now the costs have been cut down to like around $1,000 for sequencing one human genome. So that is because they have come up with these new sequencing methods like the next gen sequencing. To know more about it, you can actually look at some other YouTube videos. There are a lot of videos that explain how next gen sequencing is carried out. Okay, so now that we talked about DNA, we've talked about the human genome and why it's important, let's talk about what are mutations. And mutations, as you might already be aware, is is a word that's very commonly used these days, and when you talk about genetic disorders and this and that, mutations is something that comes to your mind early on. So what mutations are actually heritable changes in the DNA sequence of an organism. So mutation is a change which is happening in the DNA sequence, and it is heritable, which means it can be passed on from the parent to the offsprings. Okay. And the organism which possesses this altered DNA is called a mutant. So the organism which is carrying the mutation is called a mutant. And these mutations, they may or may not be detectable in the phenotype. So these mutations, they might just be there in your DNA sequence, but they do not express themselves. They're not actually um, detectable in our phenotypes, but some of them are detectable. So this is what is mutation and what causes these mutations. So there are two kinds of mutations. One of them are spontaneous mutations and one of them are induced mutations. So spontaneous mutation, as the term suggests, they occur randomly and they occur due to error in the natural phenomena of DNA duplication. So when a DNA is being replicated, so by mistake, one of the strands or the DNA polymerase might commit a mistake, and which leads to this error in the DNA duplication. And that is what is spontaneous mutations, and it is important in forward genetics. The other kinds of mutations are actually induced mutations, which are caused by external factors. They are caused by environmental factors, by the mutagenic agents that might be present in the environment. So, as you can see in this image, there are different kinds of mutagenic agents. For example, radiation, like UV radiation, X rays, chemicals, for example cigarette smokes, for example all of these chemicals like benzoyl peroxide, which is present in like sunscreens, which is present in acne products, also in skincare. Then there are these preservatives present in things like hot dogs. There are things present in barbecue sauces. So all of these are chemicals which can potentially lead to mutations. And there are these other infectious agents, for example diseases, for example HPV, the human papilloma virus, or there is Heliobacter virus, Heliobacter pylori, which is a bacteria. So there are these external factors also which can cause mutations, and those mutations are called induced mutations. Mutations that are happening naturally, that happen while the process of DNA replication, those are called spontaneous. Others are called induced mutations. So there are two ways in which mutations can occur. Spontaneous mutations are important in forward genetics, which means when you know the phenotype and you from there you want to get to know the genotype. And induced mutations are important in reverse genetics, which means when you know the genotype and you want to get to know the phenotype that it leads to. Now, coming to the different types of mutations. So there are many different types of mutations. Two broad categories of them are point mutations and chromosomal aberrations. Point mutations are actually, as the term suggests, they happen at one single point. So mutations that are happening in one or two adjacent base pairs at like a pinpoint location, they are called point mutations. And chromosomal aberrations, on the other hand, they affect multiple genes in the chromosome, or they affect the entire structure of the chromosome. So that is what is the chromosomal aberrations. Sorry. Yeah. So that is what is chromosomal aberrations. So when it comes to point mutations, there are two kinds of point mutations. One of them are base substitutions and one of them are indels. Indels, as the term suggests, is insertion deletions. So first, let's start by discussing these base substitutions in point mutations. So base substitutions are actually of two types. They're either transitions or transversions. And base substitution, as the term suggests, means that one of the nucleotide bases is being getting substituted by another nucleotide base. So at that one location, instead of one base, for example, you can see in this image, there was an A in the original one, and it got substituted by a G in the mutated one. So that's a substitution. So one nucleotide base is just getting substituted by another base. Now, these base substitutions can be of different types. One of them is called transition, another are called transversions. As you might be seeing in the image already, transitions are actually substitutions of a purine with a purine or a pyrimidine with a pyrimidine. For example, these A and G are both purines, and if A is being substituted by G or G is being substituted by A, that's called a transition. Similar for C and T. These transitions are symbolized by a small s. Transversions, are, on the other hand, are actually substitutions between a purine and a pyrimidine, or vice versa. So if an A is getting substituted by a C or by a T, if a G is getting substituted by a C or by a T, and vice versa, those are called transversions, and they are symbolized by a V. And there's something called the transition bias, which means that transitions, they occur more frequently than transversions. Or on the other, or in other words, the ratio of S by V, which is the number of transitions by the number of transversions, is always more than zero point 0.5. And this might be because it's uh, it's chemically balanced if a purine is being substituted by a purine, because of the presence of these two rings, as compared to a purine being replaced by pyrimidine, or pyrimidine by a purine, because of the change in the structure and the change in the size of the molecule, right? Yeah. So coming to what is the effect of these base substitutions. So we talked that okay, these base substitutions can happen, they can be transitions, they can be transversions. So what is the outcome? Matlab, what happens if these base substitutions are even happening? So the outcome, one of them can be a synonymous or a silent mutation. So as you might have already guessed, since we are calling it silent, so this synonymous or silent mutations, they have no effect on the resulting sequence of the protein. So these silent mutations, they have no effect on the resulting amino acid sequence of the protein. How so? How so is because, as we are aware about the redundancy of the genetic code, right, of the codons. So we know that multiple codons code for the same amino acid in the process of translation, right? So if one of these codons gets changed in a way that it's still coding for the same amino acid, then these will be called synonymous or silent mutations. Then coming to non-synonymous or missense mutations. These, as the term suggests, they result in amino acid substitution. इसमें amino acid change हो जाता है. So amino acid substitutions in the polypeptide, then that leads to an altered structure of the protein, or an altered form of the protein. अब क्योंकि amino acid change हो गया है, the protein has also been changed, and it may or may not lead to change in the functional properties of the protein. So उस protein के functional properties may or may not get affected. Okay, they may remain entirely same, or they may get affected. Okay, there are something called nonsense mutations, and nonsense mutations are ones in which the amino acid residue then gets substituted by a stop codon. So, jo codon was coding for a particular amino acid, it is now altered in a way that it is now coding for stopping the formation of the polypeptide chain. So, it's been converted from an amino acid codon to a stop codon. So this leads to a truncated protein. अब क्योंकि वो stop हो गया है the translation of the protein, it will lead to a truncated protein, and mostly a truncated protein will either be non-functional, or it will, and that is why it will get degraded. It will be destroyed by the cell, and cannot be used anymore. Okay, so there are these three kinds of effects that mutations can have. One, they could be silent mutations causing no effect, could be missense mutations leading to a change in the protein structure, could be nonsense mutations leading to truncated proteins. Okay, Now here you can see the an example of these. So in this image you can see the no mutation may, in the DNA you have TTC, which gets transcripted to AAG in the mRNA, and it's the codon for lysine. Then there are silent mutation. TTC say it becomes TTT. It codes for now AAA, and AAA still codes for lysine. So lysine, it's a silent mutation. The amino acid did not get changed. Nonsense is from ATC. It now got mutated to, so if it's ATC, so from TTC, from T, first T gets becomes an A, and now if you translate it or transcribe it, it becomes UAG, which is a stop codon. So instead of AAG, it became UAG. It's a stop codon, leads to a truncated protein. Missense. In missense, there are now again two types: conservative and non-conservative. Non-conservative matlab it is changing the amino acid, but the amino acid is still basic. So the property is more or less the same. So it's sort of conservative. So from TTC to TCC leads to AGG, which is codes for arginine, lysine, arginine. Both of them are basic. So property of the amino acid is more or less similar to each other. So that's a conservative missense mutation. And a non-conservative missense mutation is from TTC it becomes TGC, which becomes ACG in the mRNA, codes for threonine. Threonine is a polar amino acid base, and this is where the property of the amino acid is completely changed. It's no longer a basic amino acid also. So this leads to a more severe change in the protein sequence, right? So these are the different forms of point mutations and what they can cause. Now we come to indels. As the term suggests, indel stands for insertions and deletions. Insertions, as the term suggests, is जब एक extra base pair is added. So if an extra base pair is added to the sequence, that's an insertion. So for example, here you have CTGGAG. If you add this extra codon of TGG, so three base pairs got added. So this is an insertion. So not an extra base pairs in general, more than one can also be added. Deletion is when some base pairs get deleted. So for the same, it was CTGGAG. If you delete these two G's, it becomes CTAG. Then there's something called frame shift mutation, right? So frame shift matlab, now that we know that a DNA sequence is read in a certain frame of three letters each, right? DNA sequence is read in three letters each because three that forms the codons. So a frame shift mutation happens is when, so as I said, since DNA coding protein, the protein coding DNA is divided into codons three bases long. So these insertions or these deletions can actually lead to uh, can actually alter the gene in such a way that the message is no longer correctly transcribed. It's no longer correctly moved forward. So these changes are called frame shifts. Here you can see. So if you assume these three codons CTG and GAG to be two codons. So if you assume them to be say letters, for example, saying the fat cat sat, if you delete this one letter the from here, then it becomes something different only: hef, ATC, AATS, AT. So this does not make sense anymore, right? So in same similar thing happens in the DNA. Agar in the original there was something which was actually making sense, which was getting coded into the protein, now if there is an insertion or there is a deletion, it might get altered in such a way that it's no longer coding for the same protein. So it gets entirely so the frame or the reading frame in this scenario, the reading frame of it has gotten shifted by one. So if three of them get inserted or deleted at the same time, then the reading frame will stay the same, only there will be one additional amino acid or one less amino acid in the protein code. But if you delete one or two base pairs, then the frame of reading gets shifted, the frame of the codon reading gets shifted. Okay. So this was all about insertions and deletions. Insertion means addition of the base pair. Deletion means deleting the base pair, and these both can actually lead to shifting of the frame in which the codon is being read by the uh, ribosomes. Fine. Now, next thing we discussed was chromosomal aberrations and what are different types of chromosomal aberrations. So, chromosomal aberration, as we said, is कि इसमें there is no point mutation. Not like there, it's not a point mutation. Not like one singular point or one single base pair is not getting affected. The entire chromosome is getting affected, or a huge chunk of the chromosome is getting affected. So one thing to look at is there is translocation. So this here from here it has shifted there, from here it has shifted there. So this chromosome, so this might be chromosome number 20, this might be chromosome number 21. So there has been exchange in the sequence from 20 to, so that gene which is supposed to be present on chromosome number 21 is now on 20, and the gene which is supposed to be present on 20 is now on 21. So that's translocation, exchange of genes between the two chromosomes, right? Deletion means this particular gene has gotten deleted only, or this section of the chromosome has gotten deleted only. Inversion means that the gene was present in one particular direction, now it has been inverted into a different direction. So again the protein produced will not be the correct or the normal one that is present in the wild type organisms. Okay, so inversion means the direction. So if the reading frame is this way, the reading frame was this way, the gene got inverted into the opposite direction. Okay, yeah. So this is the inverted part. So it was supposed to be in this direction. That section got inverted. So you can see that it's the inverted part indicated by the arrow. Isochromosome means now it has been separated at the centromere, and these two have connected, these two have connected. Instead of this being connected to this and this being connected to this. And there are other ways. There is insertion, some extra part of the DNA sequence which was not supposed to be in this particular, just like insertion deletions we discussed in the last slide. This is insertion deletion of a huge gene or more bigger amounts of genes than of one particular nucleotide. So yeah, this is what are chromosomal aberrations. So, how do gene mutations lead to disorders? Now that we've discussed, okay, what are mutations? What are different types of mutations? How do mutations occur? And what they can lead to, like silent mutations, missense mutations, nonsense mutations. How do these gene mutations lead to disorders? What is really happening? So, genetic disorder, as the term suggests, it is a disease which is caused by changes in the DNA sequence. When the DNA sequence has been altered, it can lead to a disease which is called a genetic disorder. It can be caused by a mutation in one or multiple genes. Okay, and environmental factors can also affect the expression of genes. So, in some scenarios, as have not been discussed so far in the lectures, but in some scenarios in organisms, certain genes are expressed only when are expressed based on the environment. So only say if the temperature is above 30 degree, only then that gene will be expressed. So some of these genes, their expression is actually governed by different environmental factors. So now if that organism is present in a place which is hotter than it should be at that particular time, then those genes will start getting expressed in that organism, which can also lead to which can also cause defects in the organism's behavior. Now, there are large scale changes to chromosomes also, like changes in the number and the structure of the entire chromosome, just as we discussed in chromosomal aberrations. So, these things can cause disorders. And some of the common genetic disorders which we'll discuss now are like sickle cell anemia, which you might already be aware of. In this, what happens is the normal round RBCs get this weird shape, which they get converted into this sickle shaped cells. The cells get sickle shaped. It is an inherited blood disorder, and the RBCs, they're now sickle shaped and they lose their capacity of oxygen carrying. So they lose their oxygen carrying capacity. They will not be able to carry enough amount of oxygen, and they're not even able to pass through the capillaries properly now because they have lost their shape. So this this disorder, is caused by a missense mutation in the beta hemoglobin gene. So the mutation that actually leads to sickle cell anemia is one in which here you can see in the normal it is GAG, which codes for glutamine. But in the mutated one, it gets altered to GTG, and which in these mRNA is GUG, which codes for valine. So glutamic acid being now replaced by valine. So this one amino acid change leads to this sickle cell anemia disorder. So it is caused by a missense mutation in the beta hemoglobin chain. Missense, as we discussed, the amino acid has gotten substituted by another amino acid. Glutamic acid has been changed to valine. So it's the beta hemoglobin gene. So some symptoms of people with sickle cell anemia. Sickle cell anemia has now actually found cures, and people, not cures, but like ways to manage it, and people can live their fulfilling healthy lives even after having this disorder. But some symptoms of people that have sickle cell anemia is that they can have painful episodes which are called crisis, and they can last for hours or days. So they will experience severe amount of pain, and they can in general be experiencing fatigue, paleness in color, and they have a rapid heart rate, like the heart is pumping very fast because the organs are not receiving enough oxygen, because the RBCs are not carrying enough oxygen. And they also have this shortness of breath, because of course the RBCs are not carrying enough oxygen, means that the body is always short of oxygen. That's why we're always trying to breathe, take in more oxygen. So these are some symptoms of people having sickle cell anemia. And then there's something called genetic epilepsy. So, epilepsy is a neurological disorder which is mostly characterized by occurrence of seizures in patients. So, seizures, which are more commonly known as fits, in which the body is shaking vigorously and the person loses consciousness. So, that is what happens in epilepsy, which is a neurological disorder. And sometimes it can be caused by things like an accident or a trauma or like a head injury, but oftentimes, more often than not, it is an inherited condition. It is just inherited genetically from parents to offsprings. And one of the mutations, there are many mutations that can actually cause genetic epilepsy. And here you can see these are different sodium channels in the neurons in our brain. And there are so many of these mutations like truncation and this missense mutations. All of them can actually lead to epilepsy. So there can be a truncation mutation in this protein, there can be a missense mutation in these proteins. All of these will actually lead to epilepsy in the end. So one of these such mutation is that in a normal patient, you have this in the voltage gated sodium channel, which is called sync 1. In that you see this codon sequence CTC AAA TGG, which actually code for leucine, lysine, and tryptophan. But in the epileptic patient, the A in the middle codon gets converted to a C, and it now codes for leucine, threonine, and tryptophan, which leads to the sodium channels not working properly. And what really happens in epileptic patients is that, so as you might all be aware, in our brains we have two kinds of neurons. One of them are excitatory neurons and one of them are inhibitory neurons. Excitatory neurons are ones that fire action potential and send the signal forward to the next neurons. Inhibitory neurons are ones that actually stop these messages from passing on. So you require both of these neurons in like to be working properly. So because you can't always have these high action potential firing neurons, because you don't always require them. So what happens in epileptic patients is that the or it has been found by research that the excitatory neurons are working fine. They are working similarly to the ones found in normal patients, but the inhibitory neurons are not working properly, and so they're not able to prevent the firing of action potentials, or they're not able to prevent the forwarding of the message. And in such patients, in epileptic patients, if you perform an ECG, you see that they have a lot of action potentials being fired simultaneously. And when there is so much action potentials being firing, there kind of a short circuit happens in the brain, which is how the patient loses control, or the brain of the patient loses control over the body, and it leads to the entire body jerking and getting a seizure, while the brain actually loses consciousness. So that is what happens in this condition called epilepsy. Moving on to another disorder, which is called the Huntington's disease. What happens in this is it is an autosomal dominant disorder, which means that this disorder happens because of the dominant gene, as you can see here, capital H. So this is like a the this is the affected parent which actually has the disorder, or which actually carries the gene of capital H and has the disorder. And then this is the normal parent, and it's an autosomal disorder, which means that it is not related to sex chromosomes. So when this parent which has the Huntington's disease, they mate with a parent which do not have Huntington's disease. So because it's a dominant disorder, you don't really even a hybrid would actually show Huntington's disease. As we know, the dominant allele, it overshadows the recessive allele, and it is the one that controls the phenotype or the trait. So this capital H small h parent actually has Huntington's disease and suffers from it. And if they mate with a normal parent, then there is 50% chances of the progeny to actually develop, that the child will carry the disease, or will carry the allele for which can cause Huntington's disease, and 50% chance that the progeny will be normal. So it's an autosomal dominant disorder. And mutation in this, what is happening really is that there's a gene mutation in the gene called Huntington, which codes for a protein which is the Huntington's protein. And this gene mutation leads to aggregation of Huntington's protein in the neurons, and which leads to death of these neurons. So the neurons start degenerating. As you can see in this image, this is chromosome number 4. The mutated HTT gene or Huntington's gene is that it has 35 or more repeats of the codon called CAG. So since there are so many repeats of this codon, the protein or the mutated protein, it has an expanded region because of these repeats. In a normal patient, these repeats are around 4 or 5. So, but because now there are 35 or more of these repeats, this gene, it gets aggregated in the neurons and it leads to neuronal disintegration or neuronal death. So this is what leads to Huntington's disease, and this is how it's passed on. So if a parent which carries or which has like a capital H and a small h, it mates with a normal parent, then there is half a chance that there's 50% chance that the child might have Huntington's disease. Down syndrome, you might already be aware of this. Down syndrome is caused by the trisomy of the 21st chromosome, which means that the 21st chromosome, there are three of them instead of being just two of them. It's gain of one extra chromosome, so it is 45 plus XY. So normally we have 44 plus XY. We totally have 46 chromosomes. 44 of them are autosomal, and XY are the sex chromosomes. So it's 45 plus XY or XX. And the symptoms are, there are some distinct facial features you see here: small nose, a small mouth, and ears. So you have almond shaped eyes, and the skin folds are covering the inner corners. You have these different distinct facial features. And then you have some intellectual disability, and there is a delay in the development of the individual. Yeah. Turner syndrome. This is another. So you can see here the ones that we discussed earlier: sickle cell anemia or genetic epilepsy. These can be caused by point mutations as well. So sickle cell anemia is caused by point mutation. Genetic epilepsy can be caused by point mutation. Huntington's disease is actually caused by insertion of these codons. So of a multiple of these codons, so they're caused by insertions. Down syndrome is actually caused by chromosomal aberration because there is addition of an entire chromosome, and Turner syndrome is another chromosomal aberration in which there is loss of an entire chromosome. So it happens in this, it's a congenital condition in females in which they will be missing one X chromosome. So it also affects the growth and development of the female and leads to a short height. And you can see here that one of the X chromosome is missing. Okay. So this was all about this week that was discussed in this week. And in the last lecture, Dr. Antara Das was actually discussing about her research about transgenic model organisms and how they are used in studying human disorders, and how they in their lab actually used it. In the lab in which she was working at, they actually used it for studying genetic epilepsy. So you can go to her lecture and to know more about how that worked and how they looked at mice to study genetic epilepsy. I would just give you a brief about what are transgenic model organisms. So they're actually animals that have whose DNA has been altered in such a way so that it models human diseases, and it helps us understand the gene function. So for example, you could have mice, Drosophila or rice, um, or rats, and you could have cattle which actually are transgenic. So so which are actually transgenic and can be used to study these genetic disorders or human diseases. So what is the use of these transgenic model organisms? So in research, so they're used a lot. So they can be used to study the human disease. They're used as model for human diseases like Alzheimer's and other hereditary or genetically hereditary diseases. They are used in drug development. So new drugs and all are tested on these genetically modified organisms or these transgenic organisms, so that you can see that if somebody has this particular disease and is being treated with these drugs, if these drugs are helping or not. For example, if you're looking at mice that carry the genes for epilepsy, you can then test them for the drugs that are given to epileptic patients in order to see if these drugs are working properly or not. So that is how they help in drug development. And then you can see what are the different conditions in which these mice are getting seizures or getting affected. So that also helps you study. And as we said, because all of us share the same DNA, and the DNA has already been altered in order to mimic that human disease, so it will let you know what is directly happening or how that is happening in humans as well. And they also allow us to study gene function. They help us study how different genes are functioning and how are they affecting the phenotypes. So Uh, this is all about transgenic model organisms. This brings me to my question and answer session for this week. So we have 15 questions, and I will give you 10 seconds to answer each question. Number one, who is known as the father of genetics? Charles Darwin, Gregor Mendel, Thomas Morgan, or Watson and Crick? Which of these is known as the father of genetics? So the correct answer to this question is actually Gregor Mendel, as we already discussed. Gregor Mendel, with all of his contribution to the field of genetics, is known as the father of genetics. What organism did Thomas Morgan use for his genetic experiments? Pea plant, fruit fly, mouse, or bacteria? Which of these model organisms did Thomas Morgan Hunt use for his genetic experiments? So the correct answer to this question is fruit fly, as we already studied. Thomas Morgan had these collections of fruit flies, and he was breeding them, and he looked at the mutant with the white eyes and uh, studied how that eye color is actually linked with the X chromosome in Drosophila melanogaster. So moving on to the next question. Base substitution mutations are examples. Some examples of base substitution mutations are transitions, transversions, transactions, transfections. A and B only or A and C only. So you've got 10 seconds. So which of these are examples of base substitution mutations? Yeah. So the correct answer is A and B only. As we discussed, transitions and transversions are two examples of base substitution mutations. Transitions are ones in which purines get replaced by purines, or pyrimidines get replaced by pyrimidines. And transversions are one in which purines get replaced by pyrimidines or vice versa. Okay, next question. Mendel's law of segregation states what? So, which of these is a statement put forth by the Mendel's law of segregation? Genes are inherited together. Alleles separate during gamete formation. Genes blend in the offspring. Only dominant traits are expressed. So, you've got 10 seconds to answer this question. Yeah. So, the correct answer to this question is that alleles they separate during genetic formation. So, that is what the law of segregation is all about. Next question. Which of the following is not a part of Mendel's law? Law of segregation, law of independent assortment, law of evolution, or the law of dominance? Which of these is not a part of Mendel's laws? And so the correct answer to this question is, as you would have already guessed by now, it's the law of evolution. The law or the theory of evolution actually was actually put forth by Charles Darwin. So, Dr. Antara Das outlines one of the causes of epilepsy as a mutation in the protein SCN1A, which, as we discussed, is the um, what? So, what is SCN1A? In which a mutation happen and it leads to epilepsy. Is it a voltage gated ion channel? Is it something that prevents fevers? Which is found exclusively in intestinal cells or which is not found in mice? So, what is the answer? SCN1A is what? Yeah, so time's up. And the correct answer to this question is that SCN1A is the uh, voltage gated ion channel. As discussed in the slide, there is a mutation which happens in the voltage gated sodium channel, which is called SCN1A protein. So, this leads to epilepsy. A genetic disorder caused by a trisomy of chromosome 21 is what? Turner syndrome, Down syndrome, Klinefelter syndrome, or Huntington's disease. Which of these genetic disorders is caused by the trisomy of the chromosome number 21? So, the correct answer to this question is the Down syndrome. As we just discussed, Down syndrome is when the chromosome number 21 gets an extra copy and it leads to defects in development. Why is it useful to take a patient's family health information? So, why should doctors spend time in taking the family health information of a patient? Is it to find out if the patient's health condition is heritable? Is it to find out the pattern of inheritance, to see what country they've come from, or to understand the sex determination, A and B only, or B and E only? Yeah. So the correct answer to this question is again A and B only, to find out if the patient's health condition is heritable and to find out what is the pattern of inheritance of this disease that they might be having. So, which genetic disorder is characterized by the absence of an X chromosome? Is it Klinefelter, Turner, Down, or sickle cell anemia? Which of these genetic disorders is characterized by the absence of an X chromosome? Yeah, so the correct answer to this question, as you might have already guessed, is the Turner syndrome. Huntington's disease is an autosomal disorder, dominant disorder, which leads to protein aggregation in neurons. If a normal man marries a woman who has Huntington's disorder, what are the chances of their offspring having Huntington's disorder? Yeah. So is it 0%, 25%? It depends on whether the woman is heterozygous or homozygous, 100%, or it depends on whether the father is a carrier. So your time starts now. So the correct answer to this question is that it depends on whether the woman is heterozygous or homozygous. So from the information that's given, we can't really make out what are the chances of the offspring having Huntington's disorder because it does not tell us. The woman does have Huntington's, but we don't know if she's homozygous. She has both capital H, or does she have capital H, small h. If she has capital H, small h, then the offspring have a 50% chance of getting um, Huntington's disorder, and if she's homozygous dominant, then they will have a 100% chance of getting Huntington's disorder. So this question, the correct answer is it depends on whether the woman is heterozygous or homozygous. 11th question: Human Genome Project aimed to do what? Was it to clone human genes, identify all genes in the human genome, create genetically modified organisms, or develop gene therapy? Yeah. So the correct answer is that the Human Genome Project was aimed to identify all the genes in the human genome. So approximately how many genes are present in the human genome? 10 to 10,000, 20 to 25,000, 50,000, or 100,000. How many genes are actually present in the human genome? Yeah. So the correct answer to this question is around 20 to 25,000. Which type of mutation results in a premature stop codon? Silent mutation, missense mutation, nonsense mutation, or a frame shift mutation? So, which of these mutations results in the formation of a pre-sense or in the premature stop codon? Yeah. So, the correct answer to this question is a nonsense mutation. As we discussed, a nonsense mutation is one in which an amino acid coding codon gets replaced by a stop codon. A point mutation that does not change the amino acid sequence is called what? Silent, nonsense, frame shift, or missense. So, what does a point mutation that does not cause an amino acid sequence change called? So, the correct answer to this is it's a silent mutation. If it's not causing a change in the sequence, then it's a silent mutation. Last question is, which of the following is a chromosomal mutation, substitution, duplication, point mutation, or insertion? Which of these is a chromosomal aberration or a chromosomal mutation? So the correct answer to this question is that it's a duplication. So a duplication of a chromosome is actually one form of a chromosome mutation or a chromosome aberration. Okay. So this was all about today, and yeah, I hope you guys enjoy this video, and I hope it was informational. Thank you.